This is Between the Brackets, a MediaWiki podcast, episode 3, March 6, 2018. Welcome to Between the Brackets. I'm Yaron Koren. My guest for this episode is Mike Cariaso, best known as the co-founder of the website SNPedia at SNPedia.com, and its offshoot software, Promethease. I first met Mike in 2008, about 10 years ago, at a Semantic MediaWiki related event. Since then, we've met up on numerous occasions, usually in the US, but sometimes elsewhere around the world. Although I haven't been able to see him in the last few years, so this is also a good opportunity to catch up. Mike is also what I would call a digital nomad. He's lived in various places in the US, Europe, and Asia, most recently in South Korea for the last 2 years with his wife and daughter. I wanted to interview Mike now because I figured there was a nice tie-in with the Winter Olympics, which just happened in South Korea, but it turns out that he's already more or less moved on. He and his family do happen to be in Korea right now, but that's just sort of a coincidence because they've already started traveling through Asia. And I think it's all thanks to SNPedia that he can have that kind of lifestyle, so I guess you can call him a MediaWiki nomad too. Anyway, Mike, welcome to the podcast.
Yaron, thanks. It's great to be here with you, wherever we are.
You've spent the last 2.5 years in South Korea. What was the hardest cultural thing to adjust to?
Family, I guess. The family side of it, of my wife and my daughter and I, was, first of all, fairly new. I was only married about a year before Hannah was born, our daughter. And Hannah was only 6 weeks old when we hopped on that plane and came out here. And for the first month, I was living at my in-laws' place. And then for 1 year, we lived in a different city a little further south. And then for the second year, we moved back up here to Seoul. And we lived in a apartment just maybe an 8-minute walk away from the in-laws. And they're wonderful and they're nice people, and I'm very happy with them. I've got my wife's sister here as well. But the Korean notion of family, and maybe I shouldn't generalize to all of Korea, I can really only take one data point here, but family is a much stronger concept and more central to life. Whereas I come from an only child, single parent household, growing up in Miami, 2,000 miles away from all of the other relatives who were up in the Northeast. And so suddenly I find myself in this place where instead of being a single guy traveling when and where I want and,
right,
whatever mood strikes me, suddenly there's coordination and compromise and all that. And I think that the cultural differences of it were a significant adjustment for me. I like it. It's interesting, but I'm partly ready to move on because now's that chance to go back and just be the smaller notion of family, not the somewhat bigger form.
Right. So you're going to be traveling now. Where are you planning to end up after you're done traveling? Do you know?
I do not know. That's one of the, I guess, weird or nice things about this. I don't have deep roots anywhere. As I said, I grew up in Miami, far away from all the rest of my extended family. As soon as I went to university, my mom and had her very long-term boyfriend, who had always, actually the guy who taught me computers, they moved away to North Carolina. And so even for the university years, I didn't have a home to go to that was the home that I grew up in. It was visiting them in North Carolina and not knowing anybody else in the state. So Berkeley for a few years, the Washington, D.C. area for a few years, Netherlands for a few years, and then a lot of volunteer work in Thailand, especially on the border with Burma and Myanmar, have all sort of become home. I think my wife and I are pretty much agreed that we do want our daughter to grow up in the U.S. school system, not the Korean school system. But there's a lot of other places in the world that before she's old enough for school, I'd like to spend some time. If I have to throw a dart at the board, I'd say, Southern California, San Diego, not L.A., has been sort of nice in previous visits. And I think we did an RV trip across the whole U.S. for Thanksgiving, around Thanksgiving time. And I kind of want to do the same thing across the entire West Coast next. and sort of shop around potential homes for someday. But I think that's a few years away. In the interim, we're planning to spend some time in Vietnam in the somewhat near future. We like Australia, Sydney, or a little south of that in the not too distant future. My wife speaks Japanese fluently and grew up there to a small degree. And the chance to go live in Okinawa is definitely on our radar. And then I like the Netherlands. I'd happily go back there. I as you sort of indicated in the intro, SNPedia has been this thing I've been doing for 12 years now. For the first 10 or 11, I had other jobs and I had to work full time wherever that job was, be it the U.S. or the Netherlands or what have you. But SNPedia Promethease these days is my full-time gig, and it's just, me and a buddy, some servers inside Amazon, and as long as there's internet, I'm potentially in the office.
Sure.
So I want to use this flexibility while it's available.
Yeah. So that brings us to SNPedia, actually. Before we get into the technical details, I wanted to talk about some of the science here. The website is SNPedia, where SNP is the acronym SNP.
Correct.
What does that stand for, and can you briefly describe what it means?
Sure. I'll do a little chronology here as well. My academic, scientific, computational training. I've got a bachelor's degree of computer engineering from University of Florida, a pleasant but perhaps not world-class famous university for science or engineering. But I had done some internships and gotten experience with parallel programming in the MPI libraries in particular. And as the Human Genome Project was sort of hitting its later stages, it was becoming clear that it was a real computational problem to stitch together all this data. And they brought a couple of folks like me out to do internships and get the experience. That's where I learned effectively all of my biology. I'd taken a course or two, but it was of no interest or focus for me up until that point. But I had a PI, a principal investigator I worked for at Livermore, who was just a great teacher, mentor, friend. His name is Greg Lennon. And he introduced me to the science in a way that really made it appealing. And we liked working together. And when I finished my undergrad, I went off to go join him at a private company that he was going to then, because he was leaving the lab as well. And I just got thrown into one biology project after another, developed different sort of skills and experiences. And at that first company, he and I worked a lot with these so-called microarrays. Microarrays are it's like a piece of glass about the size of your thumbnail, and they've got a bunch of spots on them. And in each spot, there's a little bit of DNA, where the chemistry of it is going to mean that if you chop up somebody's spit properly and prepare it, you can make that person's DNA bind to that spot on the microarray. And if it matches, it'll bind and sort of light up, and you can see, yes, they have this particular pattern in the DNA.
Huh.
So the density of these microarrays got higher and higher over the years, and suddenly you were able to look at about 500,000 of these individual spots. Each one of those spots you're looking for either, is it exactly this reference pattern we expect, or is it a particular change we think might be there at one position? And so each one of those is a single nucleotide polymorphism, SNP, and in the field's jargon and lingo that became pronounced as a SNP. So these were SNP microarrays. And Greg and I had processed a lot of these samples as part of this larger company, but we got to the point where we kind of wondered about our own DNA. I think we knew things about sample J123 in the database that we didn't know about ourselves, and we were curious to explore that. So we had moved on to other companies. We weren't even working together for portions of this, but we stayed in contact as friends with a sort of complementary set of skills. He was the true scientist, and I was the guy who knew how to get the servers up and running and write the code. And we did a few other projects, and at some point we ran our own DNA on these microarrays, and we built SNPedia so that he and I at two different locations could collaborate on, hey, I saw a new publication and it mentions this particular SNP. And then we got our data back from having run our microarray, and I wrote a program to take the raw data file of my DNA and cross-reference it against the data in SNPedia and make a little report. And of course, I had to run the same tool for him, so it's a program that's reusable, and that's what becomes Promethease. But other people had all seen this trend in the science field coming along and the prices of these microarrays dropping like everything else in biology, while the density and scientific content was increasing. And not too long after we got SNPedia up and running, 3 companies on just bing, bang, boom order, literally one day apart in the case of 23andMe and an Icelandic company named Decode, both came out of the gate with these products where they were selling that, Joe Random could order this over the internet. And at that point, you had to pay about $1,000 for those 2 companies. The other company was Navigenics for $2,500. And
Wow.
Suddenly, regular people were getting the same kind of data that Greg and I had gotten about ourselves. And so it was a very easy and gradual process to just add support for the file formats that those companies started sharing their data from. And that's sort of the origins of all of this. SNPedia was a way for Greg and I to keep track of papers, but do it in the open. We thought Wikipedia was super cool. And Promethease was just a tool for him and I to interpret our own DNA against SNPedia. But these days it's a resource that basically anybody with $10 can come along and bring their data and see what you can learn.
Yeah. Well, this is great. We've already covered SNPs and SNPedia and Promethease. More clarification about SNPs themselves. As I understand it, you can consider them sort of errors in DNA.
In some cases. You and I are different people. We have slightly different DNA. It's mostly the same because we're both humans of, ancestry from common ancestors and probably Western Europe to a large degree.
Right.
So some differences between the DNA are completely normal. And there's just a handful of particular SNPs that determine eye color or hair color. And then a lot of other SNPs interact in more complex ways for the more common diseases. Things like, say, type 2 diabetes or heart disease. It's much more common for those to have many, many small DNA changes, many SNPs that are in concert, because if there was any single cause for that disease, it would have been detrimental to a big population, but instead evolution kept building more and more redundancies. There are, however, a ton of SNPs that are causes of the so-called monogenic, Mendelian, etc. disorders. There's a normal version of this gene out there. Most people have either the exact one true form or something fairly close that's mostly equivalent. But then every once in a while, somebody's born with a random mutation, and this random mutation makes that gene non-functional. And it can be a dominant mutation, and now you've got this defective version of the gene floating around causing problems. Some diseases, you need only one copy of the SNP, and it's a dominant disorder, and now you have the disease. Some of them you need 2 copies. That's a recessive disease. Sometimes it's combinations of different SNPs that cause the disease. So some SNPs are natural variation, completely harmless, no apparent difference in appearance or function or behavior or anything. Some SNPs are the basis of normal variation in a population, like hair curliness or balding or eye color or what have you. And then some are the basis of usually pretty horrible disorders. It's, I guess, one of the challenges of Promethease, in fact, is that people come to us and they expect there to be something interesting in their DNA. And if you're a normal, healthy adult, most of the time your DNA is boring, and that's really a good thing. Imagine going to the hospital, the doctor starts calling other doctors into the room saying, oh, you gotta see this, right? You're in trouble. Something's not right there. And for DNA, the same thing is kind of true. However, it's not uncommon for us to get somebody who comes in expecting fairly normal DNA and then finding, that's it. One of my favorite examples of this is somebody who, for at least 3 generations of her family, some of the members would just go blind in their 20s. One eye, and then a few weeks or months later, the other eye. And they all knew there had to be something going on, but for the most part, it didn't affect enough members of the family, and the pattern wasn't clear. Their doctors had never figured out what it was. And she came in and ran a Promethease report, and just top of the report, big red outline. I forget the name of the exact disorder now, but we said, look, you have this mutation. This mutation is supposed to cause blindness. She did a little more Wikipedia and Google for the symptoms. Everything fit perfectly.
Yeah.
And she went back to her doctor and said, I think I know exactly what this is. Now, the data she brought to us from whether it was, 23andMe or Ancestry or which company, I don't remember. It was probably 23andMe, if I recall, is not clinical grade. Anybody who brings data to us, if they bring us clinically validated, really, high confidence data, gets fairly high confidence results. But most people these days are dropping about $99 or a little bit less to get their data. And as a result, they're getting good information, but it's not reliable at the level that a doctor-ordered test would be. But often this is the first step towards knowing where to look and what clinically relevant tests to order for validation.
Yeah, and that woman, what could she do at that point, given that information?
Not a thing.
I see.
She knows what the cause is. She's able to, I guess in her case, understand that it's mitochondrially inherited, and that suggests certain things about the fact that a mother passes it to her children,
right,
but that a father does not. But there is no, in this case, there's no known treatments. There's not much research on the disorder. I mean, I shouldn't say I haven't totally studied it, but she imagine the usual scenario of your kid is sick, nobody can tell you what's going on.
Right.
Just getting a name for things is a huge step forward.
Sure.
And so she's the case I remember the best.
It's not environmental or that sort of thing.
Right. And the other one is we see a lot of cases, I read enough cases where a family has a kid, the kid is severely disabled, and nobody can explain what the cause is. We just, in the nature of the society I'm most familiar with, mothers blame themselves. They're convinced it's that, one time she stood too close to somebody smoking a cigarette, or that one time she had a glass of wine after dinner, or that's
right,
she tried LSD when she was 17. People just jump to any conclusion and they want to blame themselves, and they're terrified of whether or not having more children is. And so being able to have some explanation for what's going on and say, nope, this is random mutation, a particular photon hit the right cell at the right time. It's science. It's never crystal clear, but we can give people more information, and I feel like that's certainly a net win.
Yeah, that makes a lot of sense. One more question about the science before we move on to the technical stuff. When I was learning about this stuff, it was just about genes. We learned that there were dominant and recessive genes and so forth.
Sure.
We certainly never heard the term SNP.
Right.
So is all that stuff that we learned about genes incorrect or a simplification?
No, it's what's the right way? A gene is an article in Wikipedia. It's a sequence of characters or sequence, ATCG, ATCG, AAA, ATTT, over and over again on a particular topic. And there's mechanisms in the cell that are going to recognize a starting point and an ending point, and some rules for where to, sort of like code, there are sections that are commented out. And it's going to read this code and produce from the DNA some RNA, which gets turned into amino acids, and it all folds up into a protein. And now this protein, rather than being source code, it's sort of a compiled object, and it's floating around in the goo of the cell, bumping into other things, and sometimes these two folded proteins, like a lock and a key, they fit together, and all of the other things that we think of as, drug metabolism. How does your body take a piece of food and break it down and take a Tylenol and put something into your bloodstream? So the genes remain there as where the rubber hits the road, the reality, the three-dimensional part where the chemistry really does the things. But genes are made up of these 4 bases, these 4 letters, right. And all it takes is one change in the letters, just like a typo in a book. Sometimes the typo is utterly harmless, and sometimes the typo completely changes the meaning, right? You get to a different word, came instead of same. You change one letter and you've just changed the meaning of this sentence. Right. So the genes are there. I think the one thing that is a takeaway between maybe the high school or college version of biology, you're getting close to 40. I think you come from the same generation of science I do, is that genes were made to sound more definitive than they are. It was interesting as we were wrapping up the end of the Human Genome Project and still trying to put this all together. There was a sort of a contest among the science community. Guess how many genes are going to be in the human genome? And we only had a handful of examples, like the fruit fly, Drosophila melanogaster, and a few other organisms to look at. And everybody said, well, humans are obviously more complex than any of these other organisms. We're going to have more genes. And it's just not true. Huh. Humans are completely in the range of genes for tons of other organisms. And we are dramatically smaller genomes with fewer coding segments, fewer genes, than many other organisms. The onion that you might eat on a hamburger, its genome is massively more complicated than you. Wow. Wheat, the mistletoe that you'd kiss under at the Christmas holiday, massive genome. Huh. Salamanders, tons of other species, and we have some insight to what it is. That's sort of underlying this. But your high school notions that one gene has one effect, it does one thing, more genes means more sophistication, more complexity, is just so, at this point, naive. But it's a naivete that was shared by the entire scientific community. The estimates for how many genes there were going to be, kind of like these, there's a jar in the shopping mall. It's got a bunch of jelly beans. Guess how many jelly beans are in this jar? You win a prize. And then what they were trying to do was look for the sort of bell curve consensus across the community to sort of aim for. Because, what's the right answer wasn't clear from any side. The estimates have all proven to be about 4 times as large as reality. We have in the neighborhood of 22 to 25,000 genes, depending upon how exactly you count them. And even that is, I think, one of the other big lessons is that I come from a very computational perspective, and there are people who know the biology way, way better than me who might argue with what I'm about to say. But genes, from a computational view, are almost not a useful concept because there is so many exceptions to any definition of what a gene is. There's definitely coding segments, but there's regions upstream of the gene that act as promoters, and if they're not there, the gene is not really going to do its thing. And there can be alternate splicing of how this gene gets turned into the transcript and then the protein. And there's so many variations that computationally we need very precise definitions of things. And that's really what drew me to the SNPs early on, was that here we are talking about a one-letter change. I can model that. I can build a database of that. I can take this wonderful simplification that at this particular position, the reference says everybody has a C, and the bases before that are going to be G, G, C, T, and the bases after are going to be that. But at this one position, you're supposed to have I forget what I said, let's say G. And when we do sequencing of enough people, we find that some people have an A. And you've always got two copies, so maybe you're a GG, or maybe you're an AG, or maybe you're an AA. And that's basically the universe of possibilities. As we sequence more and more, sometimes there's one other out there, maybe all four, but it's such a small, discrete thing that you can tackle the problem computationally in a way that with the so many edge cases and fuzzy definitions, etc., genes remain difficult to grab and model and store in a database in an unambiguous way. You need so many synonyms and equivalents. And so SNPs are this computer scientist idealized view of there's one version of the code, and if you change this position in the file, it's supposed to be a 1, you make it a 0, does the code still run? Does it not still run? Does it run for 6 seconds and then crash? Does the background on the modal dialog turn from blue to green? And kind of like, AFL, American Fuzzy Lop, is a piece of source code that's used for fuzzing, randomly generating inputs of a piece of code and seeing if you can break it to detect buffer overflows and other sorts of attacks. You can sort of treat the DNA the same way. You put some cells in culture, you use CRISPR, this wonderful, I don't know whether to call it an invention or a discovery, but this new scientific technique. And we say, look, here's the normalist version of the cell. If we change this from a G to a C, does the cell behave any differently? And then you do a whole assay of what if we expose it to this chemical or that chemical? Does it live longer? Does it? You can assay and screen because the SNPs are so narrow and so specific that it gives the ability for computational thinking to attack the problem. I want to throw out, as I've been saying this, I want to throw out one more plug. I'm going to get the name wrong, but if you Google for amazing DNA, I think you'll end up at a website that has like ds9.nl in the name. And there's a Dutch guy who jumped from computation to science for a few years and started writing down all of the observations of, here's this concept I learned in biology, here's its exact mirror in source code and programming. And the parallels between code in the programmer's view and code in the DNA is code world of biology are terrifyingly, perfectly symmetrical. You have if-then-else statements. We have the lack operon. You have dynamic linking of libraries. We have... And so if somebody's hearing this, and I think your audience is much more computational, and you want to get a taste for the biology, this amazing DNA site is just awesome, and it's rigorous. He's done some nice talks at Chaos Computer Congress, the 3C in Berlin, or similar. It's really worth checking out.
Yeah. Okay. Oh, that's very interesting. I'm definitely going to check that out, and I'll put in a link to that
I
site
found it
on the
ds9a.nl/amazing-DNA. DNA Seen Through the Eyes of a Coder by Bert Hubert. I only discovered it well after I was into, sort of being a biology computer guy. But it was such a, Cliff's Notes, a few things I knew, a few things that were new, but just awesome.
Yeah. Okay, cool. Yeah, and we'll have a link to that on the site for this podcast, too. Okay, I guess this is a good point to move on to the technical stuff, which may be less interesting or more interesting. Probably less interesting to most people, but let's see. You started SNPedia in 2006?
Correct.
And even back when I met you in 2008, you were already using the extensions Semantic MediaWiki and Semantic Forms, which is now called Page Forms.
Right.
That's actually how we met. How did you start using those?
So, as I indicated, Greg and I were not sitting in an office. We were two guys in our houses. We needed to share notes. Google Docs didn't exist yet. Wikipedia was a great model for collaboration, and we knew that, hey, maybe we'll find somebody else someday who wants to share a few more notes with us. So we worked in this very open model, and we were thinking of Wikipedia in some sense for just like a note-taking app for two guys. It was obvious enough that you could, as a sort of programmer's view, well, Wikipedia is written in this PHP language code, MediaWiki. The URL is over here. I can download this. I can install this. And we did that. And what was really important, I guess, was that MediaWiki had an API. And so while we started off with two guys in web browsers clicking in text area boxes and editing text, it became possible for me to write little programs that would automate certain tasks against the wiki, be it populating new content or what became Promethease, a bot that cross-references a different text file of raw DNA versus the content in SNPedia. So pretty early on, figured out a certain structure that the SNPs would be this category in the true Wikipedia sense. And there'd be these genotypes. So the SNP says at this position in the DNA, the National Institutes of Health.gov has named this position RS1234, and this other position RS1235, and this other position RS1236.
Right.
And they define what the flanking sequence is up and downstream. So if you happen to have a somewhat different chunk of DNA, we can still talk about these things as named positions rather than needing to say chromosome 4, 1,017 positions from the start, because that's not reliable across people or across revisions of the reference genome. And I was going somewhere with this, but I think you better repeat your question. I want to make sure I get back on track here.
Just how you got into all the semantic stuff.
Okay. Okay, so we had these categories. We had SNPs, which is the position of the DNA, and genotypes. And the genotype says at that position, having an AA causes this effect, and having an AC has this effect, and having a CC has this effect. So there was, let's say, 100 pages of SNPs and potentially 300 pages of genotypes. And it was just becoming super common for anybody who took a look at this site, who we tried to talk to about it, to say, how many SNPs are in SNPedia? And Semantic MediaWiki gave me a way of making a FAQ page and having an ask statement, and it would count the number of articles in the category and spit it out as a number, and it would update itself automatically. And this literally got rid of the single most common frequently asked question. So boom, we're throwing Semantic MediaWiki into the site. Well, we need to edit all of these SNPs, we need to edit all these genotypes, and there's kind of a structure we want to have to it so that the Promethease code can parse it back out in a meaningful way. And boom, here's this page forms extension, and it makes it that much easier. I mean, I'm the geek here. Greg is no slouch himself. He's can work a grep, but he's not a programmer. And giving him forms that were attractive and easy to customize with CSS and all the rest was a nice win. And again, was meant to help open this thing up, because we're kind of staring at this mountain and saying, well, there's only 3 billion letters in the human genome, and it's really not going to get any bigger. So potentially we're looking at about 3 billion SNPs in SNPedia. The reality is not every single position is going to have an observed SNP. Even if it does, it's not always going to be interesting. But we've got an upper bound on this thing. And even with that upper bound, it's probably too big of a problem for Greg and I to tackle. But maybe it'll take 10 years. Maybe it'll take 100 years. Maybe it'll take 10,000 years. But I have no doubt that someday, if we haven't, wiped ourselves out, humanity will continue to think that understanding its own DNA is a worthwhile endeavor. and that enough people and enough time will continue to chip away at this mountain until we have done the science to explain the consequences of every single one of those letters. And so SNPedia is playing a long, long game. But even if you've only solved 0.0001% of the problem, you've probably solved a really interesting 0.0001% of the problem. So we just keep chipping away at this thing. So SNPedia knows that we've got all this work in front of us. We look at Wikipedia and say, well, they've got an infinite amount of work as well. We would like to have more help doing this. We should be doing all the things that Wikipedia has done to engage social engagement with the site. I will say with more sadness than happiness, but believe me, there's reasons to be happy about this. We have never had anything resembling the sort of community engagement that Wikipedia has had, but we haven't had none. And it means that even if only once in a while one other person comes along and adds some information to the site, great. That's one more thing Greg and I don't have to do. And we're again playing a really long game, really patient here. I think the main barrier is that we don't have a lot of contributors, but we don't have any vandals. We get the occasional bot that wants to spam, toner cartridges or something, right?
Right.
But nobody is coming in to vandalize the page that talks about how RS 678 influences cytochrome metabolism of blah, blah, blah. It's just it's so, I won't say dry because it's fascinating, but people don't want to vandalize it the way they want to say that Obama is a monkey or Trump is an orange or whatever. It's just not, it's not fun to vandalize.
Right.
So the Semantic MediaWiki solved fact number 1. Semantic forms made data entry a little bit easier and actually helped to improve the quality, because when you have anybody editing unstructured data, you're going to get more variations. Is it capital S summary or lowercase s summary? Do I put a space after it before the equals or not? And those two modules really got us pretty far along. I went to a first Semantic MediaWiki meetup in, I guess, Boston.
Yeah.
Found like, wow, there's other people using this module, and hey, almost everybody who wrote the code is sitting in this room. You were one of those people. It was fairly well timed in that I had sort of just really hit this semi-nomadic phase of life at about that time. And I moved to the Netherlands to work for this agricultural DNA-oriented company. And just like a week after I got into the country, there was a Semantic MediaWiki meetup in Karlsruhe. And you certainly know, and others in this community would know, it's that so much of Semantic MediaWiki comes out of Germany with, help me with the names here, Marcus and Denny?
Yeah, Marcus and Denny. I hope to have both of them on the podcast later also.
Right. And now, I'm sure they're active with Wikidata or related topics, but in the early days, having a foot in the U.S. where you were sort of, I think, a major anchor for development outreach community, and a foot in the Netherlands close enough to go meet up with these guys as well, was helpful for giving me the sense that I know these people. When I have a bug I've found or a feature request, it's a small, very helpful community. Maybe I'm speaking more to what it was in the early days. I can't speak to what it is these days quite as much, but I've had great experiences along the way. And at that Karlsruhe conference, somebody turned me on to, I guess, Squid as a reverse proxy to help me scale up, and later to Varnish. And outside of Semantic MediaWiki, we're all facing related issues. Spam control because of bots. But that's kind of what got me into this, and we still consider it a backbone technology. My only sort of bad thing to say is that it's such a well-solved problem that I don't have to interact with the MediaWiki layers all that often these days, because they just work. and I'm busy thinking about other parts of the technology stack that still need more attention.
Well, fantastic. You heard it here, everyone. This
It just
MediaWiki.
works.
Yeah, MediaWiki is.
There's
It's
no
very much
higher
finished.
praise in the
Yeah,
tech
sure.
industry than it
No,
just
sure,
works.
absolutely. Um, yeah, I mean, obviously still a lot of things left to do with the software, but it's great to hear that for you, it's it's
Right.
just a sort of a backbone, a back end thing at this point. Um, actually, some of the stuff you brought up before is interesting. Speaking of that FAQ number, at the moment, the FAQ says that you have about 100,000 SNPs on
About
SNPedia, which
108,000, I believe. Yep.
okay,
Yep.
yeah, I was doing some rounding. Yeah, I mean, I'd say that's pretty good for a work in progress.
Yeah, one of the things that's happened is we were early into this space, and a lot of resources didn't exist yet, and we remain sort of a outsider, third party. We're speaking to a general public audience in a fairly layman's terms. We're struggling to balance this. By far, the number one complaint we get from users or would-be users is it's too technical. I have no idea what you're saying. But at the same time, we're trying to be a bridge to scientific literature that's even more challenging. And the problem is that you can't just summarize the article, because the challenge is there's 6 different articles talking about this position. They say different things. They study different populations. How do you synthesize this down into one coherent story? But a guy in the Baltimore Johns Hopkins University for like 70 years was just the researcher who was collecting his notebooks on every single, mostly childhood development disorders that were very likely to be monogenic. You could look at the patterns of how they turned up in families and say this looks like a single gene. But his name was Victor McCusick. And he died not too long after we were into doing this, but his work in developing his lab notebooks and then sort of putting them online as the OMIM, Online Mendelian Inheritance in Man database, laid the groundwork for having well-curated databases of this kind of information. Their organization was still a little gene-centric and therefore not really applicable for what we're trying to do with Promethease and SNPedia, but it was a tremendously useful standard. They, after his death, like a lot of things, I think that the founders are different than the folks who take over, and the group behind that decided they need to work on scaling it up, getting funding, etc., but they kind of put up some licensing terms that made the data less accessible to us. It's still easy enough to see it online, but we don't have any rights to include, their summaries into what we're doing. And then other groups were affected by that. And then beyond that, the world and the technologies had advanced to a stage where there was new ways of thinking about the science. And again, the NIH, National Institutes of Health, really stepped up and started a database called ClinVar. And ClinVar has some budget and some muscle behind it, and they have really scaled up the number. What Greg and I were doing in the beginning by hand, suddenly there was a research team somewhere in the NIH producing a lot of this content. And so if you look at the graphs of our pages, you'll see these days when suddenly it just jumps. And it's bots that I've written that are scraping the content from ClinVar, very permissive licensed, direct government paid for work, and incorporating that in. But their content remains intentionally not written for a layman audience. They are a little bit, I think, cautious about people taking the information and interpreting it against their own DNA.
I see.
And so I've been able to use these bots. I write mainly in Python and sometimes Perl. These days, certainly more Python. But a couple lines of code will read their download file of what the latest version of ClinVar looks like, and I'll bulk populate that information into SNPedia.
Yeah.
And that's been true for a few other sources as well. So while a tremendous amount of sweat, particularly Greg's, has gone into the content of SNPedia, I want to offer a very big shout out to the work of ClinVar and then the bots that have helped put that 108,000 pages in place.
So how much of your data was entered manually as opposed to being imported via a bot or some such?
I mean, I don't think there's any way to make a meaningful answer to that. I'll give you the best I can. But even when the bot puts it in, the bot sticks to extremely factual-oriented things. This publication has this keyword, has this PubMed ID. And we are fairly adamant about the idea that there's two fields that we add in a structured form. One is called magnitude, one is called repute. Repute, and these are things that other groups out there have tried to do vaguely similar after us, but I think they've always kind of mucked it up by trying to find ways to be algorithmic and break it into more categories. And so we've got just these two categories. Repute. Repute is either good or bad, or it's still not set. And magnitude. And magnitude is a floating point number between 0 and 10, and it says how good or how bad is something. And I think a lot of what we've brought to the party, put on the table here, is that we're willing to take opinionated statements about complicated topics. And because we're a wiki, we're able to do it and feel like, look, on this date, this person put this information in. I now disagree with that. I'm going to change the repute from good to bad, or I'm going to reduce the magnitude. And we don't pretend that we're absolutely right in the way that a relational database does. We are comfortable living in this never finished, always improving version of a wiki database and saying, we're just going to say what we think it is now, and if we're found to be wrong, we'll change it, and there'll be a record of that. And in the sciences, it's been pretty rare to find people who are comfortable making those sorts of squishy statements. And I think that wiki and the wiki worldview has been a big influence on us in very positive ways there.
Yeah, that's a great distillation of the wiki philosophy, I guess, or as I would put it, which is, this is...
Well, I use it as a mantra.
Yeah.
Never finished, always improving.
Yeah.
And then this idea that humans do what humans are good at, bots do what bots are good at, and they work together. And those sort of four statements or two angles have been just so fundamental to what we've been doing, and I think what's unique about SNPedia. Although the second part, the idea that humans do what humans are good at and bots do what bots are good at, maybe this is a good segue, has been something I've done I've done a lot of other wiki projects, usually behind corporate firewalls or other environments. And the way that I bring that sort of approach and that philosophy to the table, to the code, to the project, I think remains something that's fairly unique and really valuable. And I think that anybody who's listening and sort of, if you're down with the wiki, realize how rare that sort of symbiosis, that cooperation between little bits of code and humans is, and how powerful it can be.
Sure. Are there researchers using SNPedia? I mean, is the data flowing the other way as well?
Absolutely. We get the best way to say is we wanted to make sure that we had one credible scientific publication so people could point to SNPedia in their own research and say, and so I forget the year, but maybe 2010 or so, we published a proper article and documented what SNPedia is. And it still gets plenty of citations. We have seen, plenty of groups when they want to do a research study on, cardiovascular disease or type 2 diabetes. they'll begin in SNP, or early on they'll use SNPedia to get a list of, okay, what are the known mutations that are relevant to this? Because it's really easy to have tunnel vision on your one topic and say, well, look, I know these 5 papers all tell me cardiovascular disease is linked to this gene. Here's a couple of papers I found about that. And to not recognize that there's some other group somewhere publishing on a topic that you don't pay attention to. Drug metabolism or something, smoking habits. Yeah. And it turns out that this group is also turning up effects on this exact same SNP. And suddenly you begin to put these 2 very unrelated topics together, and you realize that this SNP is influencing heart disease not through sort of mechanics of heart muscle formation or triggers of electrical impulse, but this one's influencing dopamine or serotonin, and it's about satiation and people's behaviors and their habits. And that's not even a great example. There's so many things where somebody thinks a particular position in the DNA is cancer related. And then you see this other class of articles that are all ending up here as well. And it's because they have this totally non-cancer effect as well. And then it's at the intersection of the 2 that you begin to gain some insight to it. Actually, I'll say one more thing there that's important on this bots and humans. I said earlier, these SNPs have these RS1234 style number identifiers. Genes have much more messy names, and they get cute names, and they get different names in different organisms, and one gene is called FTO, and another is called MTHFR, and another called TCF7L2. And the problem is those names look like words. And so FTO could be an acronym for a million different things, and it makes natural language processing or text searching quite difficult for some of those gene names. And the beauty of these SNP names, RS1234, is I don't need fancy NLP techniques to find them.
Right.
I can write a regular expression that says RS/D plus, or a few variations on that because we know how many digits, and I can grep piles of text and find these things. And so they're not used as universally as we would like, but they're used pretty widely. And it's made a lot of bits of finding and connecting literature that would be quite difficult with these supposedly human-friendly names to be very difficult tasks that are trivially easy tasks for the SNP notation. And so another sort of takeaway lesson is to pick good names, and I think a couple alphas to indicate a sort of a category, and then just sequentially assigned numerics are a super powerful, underappreciated way of doing identifiers. And I wish anybody listening, whatever domain you're in, appreciate the importance of good names and the leverage you give yourself and others in downstream processing if you pick names of this sort. Anyway, I mention that because so much of our content, if you were to go right now to SNPedia's recent changes and then hit show bots, what you're going to see is that there's a lot of articles that you could put in every single night. In fact, right now I'm looking into just the last probably 24 hours. And there's something called Oriental Bot. It used to be SNPedia Bot, but I wrote sort of a version 2.0, and it was dealing with something called orientation, about what side of the DNA is it on. Anyway, and I've got, 50 new edits, and they're just these silly little things you can click on. Again, it's a wiki. You click on recent changes, you click on diff, and you'll see that Oriental Bot found a new PubMed publication called 278-25117, title, Genetic Variants in MicroRNAs Predict Non-Small Cell Lung Cancer Prognosis in Chinese Female Population in a Prospective Cohort Study. By itself, it may be kind of a boring thing, maybe it's super fascinating, but it gets found automatically, no human has to touch it, it gets added to the right SNP page in SNPedia, and when I go to that page, there's now 30 different papers No human had to touch it. And so now I can look across all those titles and say, do they all relate to non-small cell lung cancer? Are there other studies on breast cancer? Does it only turn up in Chinese publications and Chinese populations?
Right.
Is it the leverage that we've gotten from bots scraping new articles every single night out of PubMed, bulk curation from ClinVar, means that the subjective human part, what's the repute, good or bad, how good or bad, is something that humans can add just the cherry on top. We also, another bot is coming in here and looking at published databases of how frequent are different genotypes, and it can show me whether a particular SNP is super common and found the 3 different forms in like a third of all people on the planet, or is it one of these where 99% of all people have a CC and 1% of people have a CT? And that kind of points to the idea that this T genotype must be really rare and probably really bad. Because if it's really common, it's unlikely to be really bad. Right. Because if it was, natural selection would have weeded this thing out. Instead, the common genotypes can have important effects, but they're usually parts of networks, and it's doubling your risk of this, it's halving your risk of that. It's a little more muddled. The rare genotypes are usually more high impact, high magnitude. And so it's the combination of curating PubMed, ClinVar, and these population frequency databases that are all upstream sources others produce. 2 of those databases can be done extremely mechanically. And certainly our part for all 3 is extremely mechanical. And when you synthesize these 3 things together onto one page, you get a heck of a lot of insight into the effect that this DNA change has that's not visible from maybe any of those databases independently.
Yeah.
And bots do that for us.
Yeah, I think a lot of people listening will recognize that approach of, an arbitrary increasing identifier number from its use on Wikidata.
Sure.
And who knows, maybe in the future, scientific papers and articles and so forth will have the Wikidata tag for each concept that they cover, which would be pretty cool.
I don't pray often, but that is one of the things I pray for, because I think that the people putting out the articles think in a sort of narrow view about their paper and don't have the moment to step back and think about their place in other publications in the field for the next 20 years. And if everybody were taking that long view and worrying a little bit less about the impact of their individual paper and instead making a contribution to the body of science appropriately, and then not just designing the right experiment, but doing all the things they could to make sure that their data is as accessible and as connected as possible, it is just a multiplier on human advancement in understanding disease, in understanding ourselves. Its impact is unimaginable. I'm in awe of it, and I'm sure I'm still under-recognizing all the little places it compounds.
Yeah.
And Wikidata, God bless them. I think it's I'm so proud to see the way that Wikipedia, Wikimedia, Semantic MediaWiki are all steps down this path. Wikidata is a big step forward. I suspect it's nowhere near the end of the chain, but it's a important next contribution. Tim Berners-Lee and your goals of the semantic web, I'm sorry we're slow, but we don't even know what we're doing. We're individual cells in an organism that don't recognize the larger context we have, but we are getting there. We are slowly but surely getting there. It kind of like this, looking at DNA. It might be a 10,000-year project, but it is at some level where we're headed.
Yeah. Okay. Back to somewhat more mundane topics. You already talked about Promethease, the software that that I guess you personally wrote
Correct.
that takes in people's genetic reports and compares it against SNPedia. Um, you well, let me see. I mean, you already answered a lot of my questions about it, but the current, I guess your current next step, or maybe it already exists, is is that people who run Prometheus reports can now store their genomic data on your server.
Yeah. The
Yeah, what's that about?
so historically, for the first 10 years or so, I didn't have that. There was a lot of engineering I thought was important to be responsible if I'm going to be storing genetic information. That's obviously very sensitive, potentially, like, medical information. And whether it's medical information or credit cards or email addresses, I sort of often work in this mindset where by not storing that information and remaining a boring target to hackers, you get an extra level of security that's sort of independent of any engineering you might otherwise do.
Right.
If somebody does manage to get through all these shields and layers and crypto, and well, when they get in, they wouldn't get anything, and so that makes it less worth their effort as well. But as we've done this, and we've done this for more and more people, there's value in having reference information we can compare everybody to. We often use the analogy that if you were the first person to have a telephone, it wouldn't do you much good. But when there's a million people with telephones, suddenly you can call all of these other people, and the value is there. So the same is true in trying to interpret your own DNA information. If I tell you have a C at this position, what does that mean? It's only when you look at a million people who don't have a C there, or 999,000 who have a C there and seem normal, and you find 1,000 people who all have a T there, and they all have a cleft lip or blue eyes or whatever. So the context of having other people to compare to is valuable, and that's the reason we've started to collect this information. It's an optional opt-in. Nobody is required to do it, but if you agree to let us store your data, in exchange you get free updated reports over time whenever you want. And we have your data now to compare everyone else to, which is particularly useful because the calls from different companies sometimes vary. And we'll notice that at a particular location, company X is just never calling a certain genotype, whereas every other company does it at ratios that are consistent with what we see from outside study populations. And so we're just trying to build up that information so that we have another company-specific comparison that we can use for interpreting what's real, what's harmful, what's helpful, what are the consequences. It also allows us the ability to actually go back and, well, we haven't done anything to support this yet. We might want to say, well, people with a T at this location are rare. Let's go send email to a couple of them and see if we can figure out, the literature suggests this or this. Let's see if they can confirm or deny that for us. So that's the goal there.
Yeah. Are there any new features that you want to add to SNPedia or Prometheus?
Trillions.
Oh, really?
Where to even begin with that? I'll say broadly, there's two classes of them that come to mind immediately. Is that one class of them is just things that answer common questions and confusion. Things that reduce my user support workload. The whole result of the site is tweaking it so that we get less inbound email where we have to answer the same question twice. One that comes to mind right now is that making a report for just you is a fairly simple process. People often want to compare themselves to a relative. Did I get this C from mom or dad?
Oh, I see.
And so we make a way that you can put in other files and have those other calls embedded right next to yours. But that's a different thing than making a report from mom and making a report from dad, because the way that information gets sorted and some of the, again, you get in the woods pretty quick here, but it's a different thing. And another thing we have is you might want to put other files in there because you have data from two different companies and you want to pool them both together to get a larger report with more coverage of your genome, because no company covers your entire genome. And you might have some disagreements between the different companies, and we really want to highlight where company X said you're a CC and company Y said you're a CT here.
Right.
But people are perpetually emailing with support questions where they uploaded multiple files and thought they were going to get the pooled report, or they thought they were going to get a different report. I need to find a way to make that clearer. Another one is, there was another one. When we used to be, okay, so totally independent user support topic. When we just uploaded raw data, you make a report, and we deliver the report, you're done. The universe was simple. Now we allow you to store data. People very reasonably believe that they should be able to say, well, 2 weeks ago I uploaded my 23andMe data. Now I have data from MyHeritage. I want to make a report that pools my MyHeritage data with my 23andMe data and do the same kind of comparison of where do they agree and where do they not, and what's the combined report for both of them. But I don't yet have a mechanism to let you use data you uploaded 2 weeks ago in the current report, because that wasn't part of the plumbing when everything was uploaded all in one go. And so I need to build something for that ability to pull from your upload of previous uploads. And okay, so those are just two, and they give you a sense of the kind of user support classic questions. On the other side, I mentioned this database, ClinVar. They recently introduced a new schema. The new schema is somewhat different in that it's less genotype and more allele-oriented, meaning only a single letter at a time, kinda. And the interpretation of the data is different, and they're fields that kind of relate to magnitude and repute. How good, how bad is this? Is it really pathogenic? Is it considered benign? They now break that down on a per disease category. We still kind of lump it together in an aggregate and say, is this thing good or bad across the board? If it dramatically increases your risk of, breast cancer, but it reduces your risk of cavities in your teeth, caries being the more technical term, there really are SNPs that influence how many cavities you get in your teeth. It seems like something about your immune system's fighting the bacteria a little more effectively. But if it's really bad for breast cancer, but really good for cavities, I'm still going to call that a net bad, right? But they've now broken it up, and I need to find a new way to represent their information so that we can model their information appropriately with ours. And so that's a new data source that I need to get better incorporated. Another one is we have a relatively new feature where we have the frequency in different populations. How common is this CC in Caucasians versus Japanese versus African Americans versus the Maasai of Africa? And that comes from other databases. It would be nice, well, you already have the ability to sort them and say which ones you care about the most because they're most similar to your DNA, so you can easily compare to others like yourself. I'd like to have the ability to say, well, my daughter is a great example. My daughter is 50% from my mixed Western European, mostly Irish and somewhat unknown mutt background, whereas her mother's side is pretty cleanly Korean. Now, we don't have a Korean population, but we have Han Chinese and Japanese from Tokyo as two reference populations. I would like to be able to make a report for her that says, weight the population frequencies 50% against the Caucasian, 25% against the Chinese, 25% against the Japanese, and that is the nearest approximation of her reference population. And so that's a nice little feature that maybe I'll get around to someday. So somewhere in that space is a sense of where software development is an ongoing need.
Right. So back to definitely more mundane subjects. This is a MediaWiki podcast after all, and there's a subject that's very close to my heart, which is Semantic MediaWiki and cargo extension. It's something that's come up in the previous episodes as well. SNPedia uses both Semantic MediaWiki and cargo, and they're sort of competing extensions. Could you explain why you use both and what the different strengths are of the two?
I can, but I don't think you're going to like my answer. You're a buddy of mine. You've written a bunch of modules that I found super useful. When Cargo came out, a lot of what I saw, read, heard, hit all the right buttons, and I said, yeah, that's certainly worth checking out. I don't really use the semantic layers, so let's go a little more direct. I got Cargo in there, and for me to know, docs are great, but I got to get my hands dirty and get in there. So I found some other little category of things and said, all right, I'm going to make this category cargo-based instead of Semantic MediaWiki. And if I like it, I'll just keep moving things over. But of course, there's the if it ain't broke, don't fix it sort of appeal. So I did get something working with it, and I was pleased to see that the relational database schema allowed me to go off and query it directly and get to it a little bit more simply. But I came back to you and said, I think I see a bug here. I have multiple worker machines, and they're all running jobs out of the task queue, and they are blind and independent of each other. And I think I've seen a couple cases where they're stepping on each other. They're not locking the table before they do updates. And as a result, just a handful of rows are apparently missing or duplicated or whatever the problem was. And you sort of said, yeah, well, okay. Cargo at this stage is not intending to deal with the multiple backends class of something, something, something. And that's pretty much where it's at. I didn't have any immediate need to use the information in some purpose that was, semantic or cargo tagged. And so it hasn't been a problem to leave it as is. But my impression is that, now maybe newer versions of Cargo have fixed this. I haven't really paid attention. But my build process is almost certainly pulling in the newer versions of Cargo. And to the best of my knowledge, the one category where I'm using it is probably stomping all over itself because of the nature of how I've architected the multiple backend servers serving the job queue. So now I'm going to turn this interview around and ask you. So where is that? What's up with you?
Well, I think the issues you're talking about have been fixed, actually.
Woohoo!
Yeah, but this is turning from podcast into a tech support call.
Yeah.
Yeah, I'd recommend to you and anybody else who's tried Cargo and has experienced those kinds of issues, which some people have. I'd recommend to well, you already have the latest version, but to get the latest version.
I should recheck, but it's almost 2 years since I think I was paying attention to this closely and doing that edit. So you've certainly had time for others to maybe ring that bell. And I know that I really was a pretty early adopter on Cargo. And I kind of, it was easy for me to say, well, if he's not going to jump on this one today, I'll just, no skin off my back. I don't blame you. As you heard my sort of users support horror stories or wish lists, I fully appreciate the need to fix issues in a particular order. And I was confident that if Cargo went anywhere, man, somebody else is going to be hitting you up on this one eventually. So it sounds like that day is here, and that's good to hear. I'll segue that into one more sort of related thought for me, is that, as I said, MediaWiki and all of its related pieces is been so much of a solved problem for me that
Right.
it's kind of a non-issue. I don't actually reach into the relational database or the semantic layer of the relational database to pull out values in order to do the things I do to make the Promethease reports. The reports all grab the page text and use MW parser from hell. That is the correct name of a Python library that parses wiki text. And I do all my own parsing on the fly to make the report and cache and this and that. So the only place where I've been using the semantic properties or the cargo properties is that kind of like that SNPedia fact page that tells me what's the count of how many SNPs there are. A lot of my categories of information use templates, and those templates use queries to show other things. So the SNP with the RS number queries the category of genotypes and asks for all of the genotype pages that have that RS number, and then on the SNP page it shows me all of the SNPs that are related to it. And the same is true for a couple other kinds of relationships between things. You can go to a gene and say, what are all the SNPs that mention being in this gene? Yada, yada. I've had to, because of that, I've had to do some really elaborate semantic ask statements. And I have, you call this podcast Between the Brackets. I've never had a problem with the brackets. It's the triple curlies. It's the double curlies in MediaWiki that I think are a crime against humanity in some cases, and certainly a crime against me. And I've managed, but when the Lua scripting language became an option, I was really, thank goodness, this is a nice, clean little language. It seems to have the full support of the MediaWiki team, Wikimedia Foundation,
Sure.
who otherwise sometimes does not jump on every possible technology. And I was super stoked to use Lua. But I've sort of gotten away from that. I got it good enough for my handful of needs. I'd love to replace some of my really hairy templates with Lua. Use it as a chance to get some CSS cleanups and all the rest of it. But I haven't gotten there. And so besides Cargo, the other class of extensions I really look at and want to do more with is the Lua and Lua scripting and whatever other related modules are there.
The Scribunto extension.
Scribunto, thank you very much.
Which is usually referred to as Lua, yeah.
Yeah, right. I think you could tease apart the reason for the extension versus the language versus but yes, Scribunto.
Yeah, I've seen some great uses of that are really simplified syntax.
MediaWiki as an ecosystem has been good to me personally, professionally. If nothing else, I've certainly pushed a handful. I guess I don't do too many commits myself, but I have documented an awful lot of bugs and gotten you or others in the IRC chat to say, look, here's a test case that fails. I don't have commit access right now. I'm just going to tell you guys about it, and you can get it fixed. But I do feel like I've been part of so many open source tools and packages as a involved user, and I do try to put back patches. I guess these days it's all pulls. But it's an interesting place to try to be part of such an ethos-driven community and not be 100% in that world. I think I can't verbalize that one very well. I think this is likely to be a cut part of the whole podcast because I'm so off in the woods. But it's amazing. I mean, Wikipedia just shouldn't be possible according to everything I was taught as a kid growing up.
Right.
And it exists, and it's awesome. And I've done so much with teaching people, these border zone refugees who have no resources. and showing them not just that Wikipedia gives them access to all this knowledge, but like, look, there are a couple of articles written in Burmese. There's nothing in your language yet. You can contribute to the knowledge of the world of topics. Who are the important people in the history of your people? Okay, there is an article on Bauji, but it doesn't mention this and this. You can be the one to tell the entire planet this. I just remain such a total fanboy of this world, and it's just unbelievably awesome. I'm all over the map, but this is such a rare chance for me to talk to people who care about it, and I think the same way, and to just give credit where credit's due, that be it the code or be it the content, MediaWiki and Wikipedia remain, anybody who has the religious faith, the fervor, man, I get it. I totally get it, and I respect it. It's awesome.
Yeah. Well, that's very inspiring, and this seems like a good place to end the interview. This has been a very instructive interview on a fascinating topic. So thank you very much, Mike.
Thanks, Yaron. I don't know when or where, but I'm sure we'll see each other again someday. To the rest of anybody else looking out there, Cariaso.com, Cariaso.com is me. Also Cariaso at Twitter and everywhere else just about. I'm not the MMA fighter named Cariaso. I'm the geeky one. But shout outs and all the rest are welcome from anybody who wants to dig a little deeper in any of these topics. I love talking about this, and Yaron, thanks for giving me the chance.
Thank you. This has been Between the Brackets. I want to thank my guest Mike Cariaso again for a very illuminating interview. See you next time.